Welcome students. Today we are going to start a new chapter for 6th semester student. It's an interesting chapter about the assessment and stability study and the patent. This all comes in the unit 5 of herbal drug technology. This is lecture 1 actually. The syllabus is little wider, so I need minimum of 4 lecture, and even I have seen that some question paper which is asked by KUHAS since 2021. This syllabus with 5th unit regarding evaluation of drugs by WHO, International Council of Harmonization Guidelines for Pharmaceutical Industries, patent and regulatory requirements of natural products and regulatory issues, regulations in India, for Ayurveda, Siddha, Unani, Drug Technical Advisory Board and Ayurveda, Siddha, Unani, Drug Consultating Committee, and there are many committees which we have to discuss about. And you see, this is the syllabus which is given by KUHAS according to Pharmacy Council of India. Evaluation of crude drugs, WHO, the World Health Organization, and International Community for Harmonization of Pharmaceutical Industry Production and various aspects, guidelines for the assessment of herbal drug stability testing of herbal drugs, patent and regulatory requirements of natural products, definition of the terms like patent, IPR, that means intellectual property rights, definition farmer's right, breeder's right, bioprospecting and biopiracy. These all they have asked questions actually. Patenting aspects of traditional knowledge and natural products, case study of curcuma, that is curcuma longa, and neem, regulatory issues, regulation in India about Ayurveda, Siddha, Drug Technical Advisory Board and Ayurveda, Siddha, Drug Consultating Committee, and regulation of manufacturing of Ayurveda, Siddha, Unani drugs. Schedule A, HZ means what, of Drug and Cosmetic Act. For additional schedule they have given for Ayurveda, Siddha and Unani drugs. So this is a chapter and we want to see some of the questions which they have repeatedly asked. This is from since May 2021. The question asked only in unit 5. You can see explain on patent of traditional knowledge and natural products with examples. October 2023, 10 mark question they have asked. This question you see, define bioprospecting with example. October 2023, 2 marks. 2 questions they have asked in the same year. And you can see, discuss the regulatory issue related to patent framed by various bodies in India. May 2024, 10 mark question. Breeders' right, May 2024, 2 mark question. You can expand this ASU, Ayurveda, Siddha, Unani, DTAB, Drug Technical Advisory Board for ASU, Ayurveda Siddha Unani, Drug Consultating Committee, May 2024, 2 mark question. Case study for Neem, 5 mark question, 2022, May 2022. Explain farmers' right, May 2022, 2 mark question. Explain in detail WHO guidelines for the state assessment of herbal drugs, May 2021, 10 mark question. Case study of Curcuma, May 2021, 5 mark question. Define patent and IPR, intellectual property rights, May 2021, 2 mark question. Schedule Z for 2 mark question in May 2021. Describe the WHO and ICH guidelines for stability testing of herbal drugs. 10 mark question they have asked. July 2023. Discuss briefly about patenting and regulatory issues of herbal drugs, July 2023, 5 mark question. Explain breeders' right, July 2023, 2 mark question. Explain again the abbreviation ASU DTAB DCC, they have asked July 2023, 2 mark question. Define IPR, again repeatedly they have asked in July 2023, 2 mark question. Intellectual property rights. State International Community of Harmonization for Pharmaceutical Industry Guidelines for evaluation of herbal drugs for WHO and ICH, both together I have given. So you can write there is 5 mark question, July 2022. Define Schedule Z, repeatedly asked, January 2022, 2 mark question. Define Bioprospecting, July 2022, 2 mark question. Explain the benefits of patenting, January 2022, 2 mark question. Discuss one case study of Curcuma longa and on case study, not one case study, on case study of Curcuma longa and Neem related to patent for natural products in India. So you have to write 10 mark question, detailedly they have asked. Briefly discuss on stability testing of herbal drug, February 2024, 5 mark question. Farmers' right, February 2024, 2 mark question. Define Schedule Z, February 2024, 2 mark question. Enumerate the term patent, December 2022, 2 mark question. Define biopiracy, December 2022, 2 mark question. You see, 26 questions have been asked from since 2021. I have taken only 8 questions they have asked in KUHAS. In 8 question paper, 26 question means you just imagine minimum 3 and above questions have been asked in 1 question paper. So you have to study this chapter very clearly, detailedly, and make yourself comfortable to answer the questions in the final exam. So we will start today's class little, as I have taken some time for the discussion of the question paper. It may take little time. Be patient to listen to this class. If it is little lengthy, but listen to the class, at least spend 40-45 minutes. You can listen and it will be helpful for you to write. I cannot cut short my classes because it needs some explanation and details, which is already done. These all of them actually guidelines for assessment of herbal drugs by WHO and ICH, Harmonization of Better Health, that is done by International Community of Harmonization for Pharmaceutical Industry in interest to the health of the human being. World Health Organization, together they have given, that is this International Council of Harmonization of Technical Requirements of Pharmaceutical Industry or Pharmaceuticals for Human Use is the correct abbreviation for ICH. An assessment, evaluation, standardization of the drug means confirmation of its identity and determination of its quality and purity and detection of nature of adulterant by various parameters. So the purity and the truthfulness of the drug supply of the herbal drugs should be identified, like morphological characters, microscopical characters, physical characters, chemical and biological observations. The evaluation of herbal drug is necessary because of three main reasons have been given by WHO and ICH. What are the three main reasons? The first one is every herbal drugs or herbal products, biochemical variation in the drug. Different countries, different place, production, different types. They have different biochemical variation in the constituents, size and types and quality is different. So you need assessment, evaluation, standardization of their preparation and supply. Number two, deterioration due to improper processing and storage. The processing technique and the storage techniques will be differ from country to country, place to place, person to person, and industry to industry, and place to place. So that should be completely controlled by means of that. That may vary the constituents quality and the efficiency. And number three, that is C, is adulteration and substitution. There are fraudulence, intentionally, non-intentionally, adulteration, substitution, and the quality may come down. So assessment, evaluation, standardization should be done for herbal drug technology, for herbal drugs and the products. This was given by guidelines by WHO and ICH. Very clearly the students can write these points. So first, this is just a picture to show you biochemical variation of the drug. You can see the pictures, variation of the plants in the color, in the size, constituents from different places of the size. Places. Deterioration due to improper processing and storage. Just a picture of a fridge is shown. So storage may be refrigerated, maybe some other means of the storage will be different types of products. It also having influence. Adulteration, you can see an example, just an example I have shown, like strychnine, or which is present, you can see Strychnus naxomica can be adulterated or can be substituted by Strychnus potrotum, which contain less quantity of strychnine and brucin. So constituent differs. Just an example. Herbal drug formulation, according to WHO, what is herbal drug formulation? What is herb? A herbal drug or a formulation is regarded as finished labeled product that contain active ingredients such as aerial part or underground part of the stem or any other plant material, or in combination thereof, whether in the crude state or in the plant preparation or in the plant formulation or in the galenical preparation. It is according to WHO, this is the definition. Guidelines for quality control of herbal formulation. WHO, World Health Organization, has given certain guidelines for assessment of herbal drugs, and most of the countries have adopted these guidelines. The following are the aim and the objective of WHO guidelines and standardizing the herbal drugs. Quality control of crude drugs material, plant preparation and finished product. First, their objective. Stability assessment and shelf-life of the herbal product and the herbal drugs. Safety assessment, documentation of safety based and or on experience on toxicological studies. Assessment of efficacy by studying the pharmacology of drugs and evaluating their biological activity. This is also very, very important point. So you can see standardization of quality evaluation of the herbal drugs. This picture can give you the complete chapters detail, complete lecture, today's lectures detail. You can see first one is botanical identity. In that you can see macroscopic, shape, external, marking, authentication. All this comes in a botanical authentication. Approval should be taken for any plant for a research. Macroscopical identity, shape, external, markings, all these things. Microscopical, when you see qualitative, quantitative microscope, qualitative microscope, number qualitative, like stomatal index, qualitative, quantitative, lycopodium spore method, width of the, some quantity, how many numbers. SEM studies, that means scanning electron microscope, enlarged size studies in a minute particles, calcium oxalate crystals or starch grains, minute or aileron grains, which can be studied very detailedly. Power studies, powder microscopy, section, TS, LS, all these things. Then comes physical evaluation, which you studied in your 4th semester, moisture content, extractive value, acid value, fluorus analysis, and even foam index and swelling index can be coming. Nowadays it is in pharmacological or biological study it comes, because they are having some biological activity. So you can see chemical is a very important and a wide evaluation, where you can see qualitative, quantitative. In qualitative, HPLC or HPTLC, high performance thin layer chromatography studies, can be even fingerprinting study, secondary metabolite study, DNA fingerprinting studies. And in quantitative studies, you can see HPLC, HPTLC, GLC, gas chromatography can also be used, chromatographic techniques. Radioactive contamination is a special study should be done if there is any type of plants which is available, like any type of nuclear power plants are there, that type of contamination may comes to our products. So when you come to biological study, you can see microbial contamination, pharmacological contamination study, and toxicological study. In microbial contamination study, total viable aerobic count, aerobic bacteria count can be determination of pathogens, determination of aflatoxins content. The toxin, exotoxins can be also studied, which can be seen, and also pharmacological bitter value, hemolytics activity, even foam index, swelling index comes in the pharmacological biological study, and toxicological study, determination of pesticide content, very important, and heavy metal which is present. These all studies should be done. Bacterial study, fungal study, these all studies can be done. And I did not tell about organoleptic. The initial one is organoleptic. I should have started from organoleptic, then botanical, physical, chemical, and biological. Organoleptic, color, odor, taste, texture, and fracture. First you have to study any product, and authentication. So you see, this is the full chapter and full details about the standardization of quality evaluation of herbal drugs by WHO and ICH, why they have given, why you need standardization, and what all different standardization can be. Simply a student can write in detail. So now we will see in some details, not going in very detail, but I have to give some definition at least, which you have already studied in 4th semester, but it's my duty to give you some idea for writing the exam. You have to listen and you have to write. So you see, and you have to make yourself notes also, little wider you can research and write. Quality control standardization of herbal drugs formulation according to WHO and ICH. The first one is authentication of herbal drugs. Second one is organoleptic macroscopic evaluation. Third one is microscopic evaluation. Fourth one is physical evaluation. Fifth one is chemical evaluation. And then seventh one is biological evaluation. And again we have not, seventh one is biological evaluation. Sixth one I added from the chemical evaluation. One more thing is, that is chromatographic instrumental techniques. Chromatographic and instrumental, they also comes in the chemical evaluation, but we have advanced technology like HPLC, HPTLC, DNA fingerprinting studies. These all have come to play in the phytochemical investigation and the standardization of herbal drugs and formulation according to WHO and ICH. So you see, fifth one is chemical evaluation. Additionally, we are adding that chromatographic and instrumental techniques. Different techniques we have to study. Basically, if the students are interested, even in my There are lectures specially for HPLC, HPTLC, DNA fingerprinting, specially alone classes are there, lectures are there, you can listen to that. And biological evaluation, there are many like microbiological evaluation, pharmacological evaluation, and we can go for even toxicological evaluation. First, authentication of the drugs, very important. The existence of numerous plants in the world, species, and some species make it difficult to properly identify them. So hence, it is essential that before starting any research or any process of herb, they need to be properly identified and authenticated from a reputed institution or organization. This will be helpful for all the students, even the 8th semester student also should listen to that, how to get authentication, how to start my research with a herbal or with a plant. The following institutions are involved in the authentication of herb. Name of institution, Central Council of Research in Ayurveda and Siddha, CCRAS. Central Council of Research in Unani Medicine, CCRUM, Central Council of Research in Homeopathy, CCRH, and Central Council of Research in Yoga and Naturopathy, CCRYN, Central Council of Indian Medicine, CCIM, Central Council of Homeopathy, CCH, laboratories like Pharmacopoeia Laboratory of Indian Medicine, PLIM, Homeopathy Pharmacopoeia Laboratory, HPL, and national institution like National Institute of Homeopathy, NIH, National Institute of Ayurveda, NIA, National Institute of Unani Medicine, NIUM, National Institute of Naturopathy, NIN, National Institute of Siddha, NIS, and Institute of Postgraduate Training and Research in Ayurveda, IPGTRA, and Rashtriya Ayurveda Vidyapith, RAV, and Morarji Desai Institute, National Institute of Yoga, MDNIY. Any institution you can use for this authentication. Number 2, organoleptic and macroscopic evaluation. Visual inspection provides the simplest and the quickest means to establish identity, purity, and the quality of the herbal drugs. A sensory or organoleptic evaluation describes color, odor, taste, consistency, fracture, surface of bark, shape, texture, and appearance of drug are evaluated. Number 3, microscopic evaluation, in which there are two. One is qualitative microscopy, second one is quantitative microscopy. In qualitative microscopy, stomatal number, stomatal index, palisade ratio, vein islet number, and in quantitative microscopy, Lycopodium spore method, length, width, number of fibers, and length, width, number of trichomes. These all you have studied in your 4th semester. Utilize all the things procedure, very, very important. Number 3, that is microscopic evaluation. Number 1, what we have discussed is qualitative microscopy. It is used to identify the herbal drugs by studying its microscopical characters such as transverse section, longitudinal section. RLS is also there. Radial longitudinal section, powder microscopy, leaf constituents, trichomes present, stomatal index, and leaf constants including the dimensions of palisade ratio, stomatal number, stomatal index, vein islet number, vein islet index is also there, which can be studied. These are some of the pictures which I can say that is you have done many. In 5th semester you have done TS also. So you can see stomatal number, index, the difference, detailedly you can write by means of your basic knowledge which you studied before semesters. The stomatal number is an average number of stomatal cells present in the square millimeter of epidermis. Stomatal index is the percentage number of stomata formed by the total number of epidermal cells including the stomata being counted as one cell. Palisade ratio, it is an average number of palisade cells beneath each upper epidermal cells. That's a picture which shows about the palisade, presence of palisade cells and the stomatal are presence there. Vein islet number, even veins also can be seen very clearly in the picture. It is number of veins islet per square millimeter. Area of leaf calculated from the center part of lamina between the midrib and the margin. Now the quantitative microscopy, Lycopodium spore method, which is used for Lycopodium spore because it is uniform in size, so number of starch can be identified. It is used to evaluate the quantity, amount of number present. Example, Lycopodium spore method to determine the amount of starch, grains and length and number of fibers, trichomes present also can be identified by means of stage micrometer and eyepiece micrometer. Even you can do for quantitative microscopy, which you have done in 4th semester, is also included in this quantitative microscopy. Now comes the physical evaluation. This also you have done completely study in your 4th semester, in which you can see determination of foreign organic matter, determination of ash value, determination of extractive value, determination of moisture content and refractive index. This any students can write very clearly. What do you mean by this determination of foreign organic matter? Drugs should be free from mold, insects, animal feces, matters and other contamination such as earth, stones and extraneous matter. Foreign organic matter should not be not more than 2% weight by weight. So determination of ash value, we know that this is the muscle furnace or insertion can be done by means of making it more than 600 to 700 degrees centigrade, and you can see insertion can be done. So this furnace, you can use this residue remaining in after insertion in the ash content of the drug, which simply represents inorganic salts naturally occurring in the drugs or adhering to it due to adulteration can be identified. 3 types of ash value you might have studied. That is, total ash value is designed to measure the amount of inorganic impurities present in the crude drug. The drug materials is subjected to incerate in temperature about 500 to 600 degrees centigrade. Remove all the carbons. Total ash usually consists of carbonate, phosphates and silicates and silica. So you can see that acid insoluble ash is residue obtained after extracting the total ash value with HCl, dilute HCl. It gives an idea about the earthy matter present in the drug. Water soluble ash. The total ash content which is soluble in water is known as water soluble ash. It gives the idea about the presence of water soluble salts present in the drug. Determination of extractive value you might have done by means of extracting, even by means of simply extracting by the test tube or by means of Soxhlet apparatus can be do. It's an idea, it gives an idea amount of chemical constituent present in the drug. Extractive values again sub-classified based on the nature of the constituent present in the drug as water soluble extractive, alcohol soluble extractive, volatile ether soluble extractive and non-volatile ether soluble extractive values. So determination of moisture content. Determination of moisture content is very important to know. About 10 grams of drug is taken and it is in an evaporating dish or a china dish. Then it is dried in 105 degrees centigrade and 3 hours and weighed again. The drying of weighing continued for an hour intervals until difference between 2 successive weighing corresponding to not more than 0.25%, then it will be taken as consideration. You will use hot air oven, desiccator and weighing balance. The reading is taken after a constant weight is reached in the moisture content is determined. Refractive index sin I by sin R. So you know that it is an Abbe refractometer which is available in our lab, and digital Abbe meter is also nowadays available. The refractive index can be easily given by sin A by sin R if it is a fixed oil. Can be even found out. When a ray passes through one medium to another, we know very well, difference in density, it bends to the original path. So that's why sin A by sin R will give the refractive index. Thus the ratio of the velocity of the light in the vacuum and the velocity in the substance of the light passing through terms as a refractive index of the second medium. So depending upon the purity, it is constant for the liquid and can be considered as one of the standardization procedure and data. The chemical evaluation, you can see it consists of qualitative and quantitative method. Qualitative chemical evaluation, qualitative evaluation, chemical test comprise of various chemical tests to identify the natural compound present in the crude drug. Examples, like we might have seen that I have given very simple 1 or 2 examples. You have studied many preliminary phytochemical investigation, chemical tests should be mentioned here if they are asking chemically how you will evaluate. So test for alkaloids, Mayer's test, drag and draft test, Hager's test, Wagon test, etc. Glycosides, bone tracker test, modified bone tracker test, and Molish test, Baljet test, legal test, Killer-Kilani test, etc. Test for phytosterols, Lieberman-Bouchard test, and test for tannins, phenols, ferrichloride test, and test for proteins and amino acid, Miller's test, Burette test, Ninhydrin test, test for gums and mucilage, swelling index test. Quantitative for chemical evaluation, quantitative test for chemical assays, chromatographic method which are used to quantity of chemical compound present in the crude drugs. So chromatographic techniques, when you come to talk about, there are many techniques which you have already studied in the 5th semester and even in that thing you have to apply. If I have given only a small details about each chromatography, just a definition, but if you want to interested, you can have separate classes available in my channel. You can listen to that so that you will increase your knowledge and you revise your knowledge what you studied in your 5th semester. The chromatographic techniques are new and the most common method used for separate, identify and quantify the plant constituents. It consists of various methods. As we know that TLC, paper chromatography, very commonly we are used for amino acids, and HPLC, HPTLC, column chromatography, GLC, gas liquid chromatography. Thin layer chromatography, we can see that is very interesting, which you have done in the lab also. Thin layer chromatography techniques becomes the most important analytical for separating and determining of natural products. It is a simple, economical and rapid method to analyze plant extract and carry out the fingerprinting of plants, the constituents, by using the standard marker compound or a standard compound which is useful for comparing with the test sample. And high performance thin layer chromatography, HPTLC, is the advanced technology from the TLC. It is one of the very useful method for the quantitative and qualitative analysis of plant extract. It is an advanced technology, advanced form of thin layer chromatography, TLC, with shorter time and precise result. High performance liquid chromatography, HPLC, one of the most versatile, safest, dependable, fastest and sensitive chromatographic technique for the quality control of the drug. The term liquid chromatography refers to those methods where separation takes place in the packing column which acts as a stationary phase. You know that partition chromatography. A mobile phase is used as an eluent, and the HPLC mobile phase is forced through the column through high pressure. So if the student is interested to know about HPTLC and HPLC, separate class is available in my channel. You can listen. Gas liquid chromatography. Gas liquid, most selective and versatile form of gas chromatography. All compound cannot be separated by this one, and you can use that gas very fast are very interesting, which is even if it is connected with mass spectroscopy to find out, identify certain compounds. And commonly it is used as an assay and analysis of starting material and drug substances, quantification of the drug substance in formulation and assay impurities and solvents in drug substances. Column chromatography is the old technique, the initial technique for the chromatography. Large quantities are separated previously. Based on the liquid chromatography, it is which mobile phase is formed of liquid phase over a stationary phase packed in a column. The column is made up of either glass or metal. It is oldest method and the most common practiced method for isolation of pure compounds. Biological evaluation. In that we should, as I told in the beginning, we have pharmacological evaluation where we have to know about toxicological evaluation and microbiological contamination study. In pharmacological evaluation, we can go for bitter value, swelling index, hemolytic or hemolytic, the lysis of hemolytic activity, foam index, and when you come to toxicological evaluation, determination of pesticide residue, determination of heavy metals, arsenic and other heavy metals can be done, and microbiological contamination, determination of microbes, and aflatoxin content determination, and lastly, we should not leave that radioactive contamination. This is what we are going to discuss today. A small introduction for every test, and we will just go ahead. A simple class which I am taken, but it may be lengthy because there is some need which already we have studied in your 4th sem and 5th sem. Please revise your knowledge with little more and prepare your own notes. Bitter value, medicinal plants, materials, they have strong bitter taste, bitters. They are called as bitters, are employed therapeutically mostly as an appetite agents. Their bitterness stimulates the secretions of gastrointestinal tract, especially in gastric juice. The bitter property of the plant material are determined by comparing the threshold bitter with which you are standard comparing concentration of extract material with a dilute solution of quinine hydrochloride. That is the standard which you used. The bitter value is expressed in units equivalent to the bitterness of the solution containing 1 gram of quinine hydrochloride in 2000 ml water. Bitter value calculation is unit per grams using the following formula. That means 200 into C by A into B. 200 into C divided by A into C. So the A is the concentration of the stock solution, milligrams per ml. B is the volume of the test solution in the test tube and threshold concentration. And that is equal to the volume of the quinine hydrochloride R. That means R means reagent in milligram in tubes with the threshold bitter concentration. Then you can see next one is hemolytic activity. Many medicinal plants, materials of families like Caryophyllaceae, Ariliaceae, Sapondaceae, Perimylaceae and Dioscoraceae contains saponins. They are having a property of hemolytic property. So the most characteristic properties of saponin is their ability to cause hemolysis. When added to the suspension of blood, saponin produces changes in erythrocyte membranes, causing hemoglobin to diffuse into the surrounding medium. So the hemolytic activity of the plant material or the preparation containing saponin is determined by comparison with the reference material saponin R. That means saponin reagent, which is standardly given by WHO, which has an high hemolytic activity in 1000 units per grams. So the hemolytic activity of the medicinal plant is calculated using the following formula: 1000 into A by B. The 1000 is the standard number which is taken from the standard saponin R, where 1000 is defined as the hemolytic activity of the saponin R. The standard is related to the ox blood. A is the quality of the saponin R reagent that produces the total hemolysis in grams, and B is the quantity of the plant material that produces total hemolysis in grams. This you can see the picture how it can be given for Moringa seed extract. They have seen the hemolytic activity, the blue, red, green, which is given very easily about the quantity in 12.5, 25, 50, 100. And you can see the picture here, the complete hemolysis, how it is shown in the color, and partial hemolysis and no hemolysis, the color we can see very clearly. So the absorption in the UV spectroscopy of the light passing through the solution is the graph which is shown in this picture. And now swelling index is done by means of using measuring cylinder. The product will be supplied in a powder form or the material and water will be added. The swelling index is the volume in ml taken by the swelling of 1 gram of the plant material. Whether it is bentonite or whether it is plant material means or an ispogul or linseed, taken up the swelling by 1 gram of the plant material under specific condition. Its determination is based on the addition of water or the swelling agent as specified in the test procedure of the each individual plant material, either whole or cut or pulverized. Foam index, you can see the foam, frothing foam, 1 centimeter concentration which is required to form will be calculated and the foam index can be determined. You can see they will different concentration and which one is giving 1 centimeter height and that concentration is used for the calculation. Many medicinal plants material contain saponin that can cause a persistent foam when an aqueous decoction is taken. The foam ability of an aqueous decoction of the plant material and their extract is measured in the terms of foam index. Foam index is calculated by using the formula index 1000 by A. What is this 1000? What is this A? Where A is the volume of the ml of the decoction used to prepare the dilution of the tube, that which give the foam of height 1 centimeter is observed persist for some time. Determination of pesticide residue. We can use pesticide residue by means of various instruments. Emission spectroscopy can also be used. That's why this picture is shown. WHO and FAO, Food and Agriculture Organization, set a limit for pesticide which are usually present in the herb. These pesticides are mixed with the herbs during the time of cultivation. Pesticides like DDT, BHC, DDT means dichloro-diphenyl trichloroethane, and BHC, benzene hexachloride. Texaphene, aldrin causes serious side effect in human being if the crude drug is mixed with these agents. So the average pesticidal residue should not exceed more than 1%. The average pesticidal residue limit, ARL, is the symbol in milligram of pesticide per kilogram of plant material, can be calculated on the basis of maximum acceptable daily intake of the pesticide for human being, ADI, which is recommended by World Health Organization, and the mean daily intake, MDI, of the medicinal plant material. ARL is equal to, that means average pesticidal residue limit is equal to acceptable daily intake into EX, where is EX 60 divided by mean daily intake, and ADI we know that maximum acceptable daily intake, which is given by WHO pesticide milligrams per kg according to the body weight. E is the extraction factor which determines the transition rate of pesticides from the plant material into the dosage form. MDI we know that mean daily intake of medicinal plant product. Determination of pesticide residue, again one more points which is added. In the field of chromatographic research on effort are being applied the target high throughput analysis. Sometimes you should know what is HTA, high throughput analysis of polar pesticides by a single multi-residue method. A triple, quadruple mass spectroscopy or mass spectrometer attached with gas chromatography, gas GCMS, or a liquid chromatography HPLC connected with mass spectroscopy platform is identified for quantity their residue with high sensitivity and selectivity and sensitivity can be identified using this new technology, new instrument of GCMS. You can see this is LCMS instrument are available. Just I want to show the picture. Just you can see this picture. It is liquid chromatography HPLC connected with mass spectroscopy. Determination of arsenic and other heavy metals is also one of the important toxicological study which has to be performed. Contamination of medicinal plants material with arsenic and heavy metals can be attributed to many causes, including environmental pollution and traces of pesticides. The content of lead, cadmium to be determined by inverse volumetric and atomic emission spectroscopy. You can use atomic spectrometry like flame photometry. It is also flame photometry having a small principle be related to that. Emission spectroscopy. The following maximum amount of dried plant material which are based on the ADI values, maximum acceptable daily intake of pesticide values are proposed. ADI is equal to maximum acceptable daily intake of pesticides milligrams per kg of body weight. Lead 10 milligrams per kg, cadmium 0.3 milligram per kg. This is atomic emission spectroscopy. Just to show you the picture, there are many details are available. Even my separate lecture is also available. Determination of microorganism. This is about the microbial evaluation. Usually medicinal plants contain bacteria, mold, and coming from soil and atmosphere, analysis limit for E. coli and molds clearly thrown light towards the harvesting and production practice. So there is a limit is given by WHO. The substance known to be even aflatoxin will produce serious side effect. It consumes along with the crude drug. So the following tables used to give the maximum microbial allowed in the raw material and finished product. You can see E. coli 10 to the power of 4 raw material and 10 to the power of 1 in the finished product. Salmonella should be nil in the raw material and should be nil in the finished product. And total aerobic bacteria 10 to the power of 5 and also finished product it should be nil. In raw material enterobacteria 10 to the power of 3 but finished product it should be nil. Determination of microorganism as shown in the picture. You can see the colony pictures like this, and it is possible for you to do by means of agar nutrient medium, and you can find out if there is what type of organism is present and what percentage it is present. Aflatoxin content. There are many aflatoxins, B1, B2, aflatoxin B1, aflatoxin B2, aflatoxin M1, aflatoxin M2, aflatoxin G1 and aflatoxin G2 given in the structure. And these all present in certain value, and aflatoxins are naturally occurring mycotoxin produced mainly by Aspergillus, that means Aspergillus flavus and Aspergillus parasiticus. So these two organisms, the presence of aflatoxin can be determined by chromatographic method using standard aflatoxin B1, B2, G2 and G2 mixtures. Indian pharmacopoeia method, IP method, not more than 2 microgram per kg, and aflatoxin B1 and total aflatoxin 4 microgram per kg. And United States America or United States pharmacopoeia USP method, not more than 5 ppm parts per billion of aflatoxin B1 and total aflatoxin 20 parts per billion. So this is the limits they have given according to WHO. Finally, we should know about the radioactive contamination also. Sometimes it is possible, it is dangerous also. Microbial growth of the herb is usually avoided by irradiation. This process also sometimes cause the impurities. May sterilize the plant material, but the radioactive hazards should be taken into an account. It can be sterilized, but at the same time, industries nearby, nuclear power generators are there. It may affect our product, our plants, agriculture products. So the range of radionucleosides that may be released into the environment as a result of nuclear accident might include long-lived and short-lived fusion product. You should know this name: acetinides activation product, actinides. Actinides are elements with atomic number from 90 to 103. Following elements: actinium. They include naturally occurring elements like thorium, protactinium and uranium, and 11 transonic. Example, artificial producing nuclear reactions. Nevertheless, all actinides are radioactive. So it is necessary for you to study. The radioactivity of the plant substances or samples should be checked according to the guidelines given by International Atomic Energy in Vienna and that by WHO. International Atomic Energy in Vienna is the institute of the picture which I want to show it. That is International Atomic Energy Agency, was established in autonomous organism on 1957. Though established independently of United Nations through its own international treaty, the IAEA, that is International Atomic Energy Agency, reports to both the United Nations and General Assembly and the Security Council. The IAEA, that means International Atomic Energy Agency, is the headquarters in Vienna, Austria. This is just for an general knowledge to know. I have just added. Thank you for attending the class. You make yourself comfortable, and also you just refer. I want to make yourself a notes and study, and we'll have continuous class in this chapter, because I have given the question papers they have asked. Very interesting chapter, very important chapter. Like how we have studied about the nutraceuticals, this chapter also, if you study, you will get at least 3-4 questions in that and 3-4 questions here. So some 50% of the questions can be easily covered by these chapters. Thank you.